brain, and that this is critically important for nervous system function. And so the story, the first story that I'll tell you starts with a former graduate student, Kaylee Hogan, who identified a putative tRNA methyltransferase as a candidate synaptic gene. And so she found that this had this expression pattern that I mentioned. When she knocked it out, she saw synaptic growth phenotypes. And what it does in yeast is it is an enzyme that methylates wobble uridines in the anticodon region of tRNAs. There are 2 in animals, and I'm actually not going to tell you about this one, CG42261, which is the homolog of TRIMT9B in humans. And this we're wrapping up. This should be on bioRxiv in the next week or 2. I'm going to tell you actually about our next story, which is about ALK-BH8, which is the paralog, the animal paralog of TRIMT9B. So methyltransferases, there's about 11 in yeast, and this has expanded to nearly 30 in animals. So this has been a big expansion of this family. And so this gene is known as CG17807 in flies and ALK-BH8 in humans. Okay, so just a quick reminder in case you're like me a few years ago and haven't thought about tRNAs for a while. So basically, tRNAs decode mRNAs by binding to codons and bringing the corresponding amino acid to the growing polypeptide chain in the ribosome. Wobbles, sorry, I should talk about wobbles a little. So the wobble position is a position that can either bind the cognate nucleotide, or it can bind other nucleotides depending on, it turns out, such things as post-transcriptional modification of the anticodon in the tRNA. So tRNAs, yeah, you might think that they're housekeeping genes and that they're just everywhere and they just do their job, but it's not true at all. So first of all, there are many tRNA modifying enzymes, including tRNA methyltransferases, that modify tRNAs. tRNAs are the most heavily modified RNA in the cell. Over 100 modifications have been identified on tRNAs. And many of these genes are starting to be associated with neurological disorders. So these have been heavily studied for the most part in yeast and other single cell organisms. But we know, for example, that NSUN2, TRIMT1, that variants in these both lead to intellectual disability. And so clearly they have important nervous system functions. And so what does methylation of tRNAs do and other post-transcriptional modifications? It's important for the stabilization of tRNA structure. It's important for the fidelity of translation, so for the recognition of the correct codons in particular. And it's important for codon-anticodon interaction. And in the case of wobble uridines, position, modulating whether or not the anticodon can interact with only cognate codons or with wobble codons. And so you can see how that would lead to differential translation of mRNAs with different codon content. And so that's actually been shown in yeast for TRIM9, that under stress conditions, it's important for altering which mRNAs are translated. And so we were very surprised to find a tRNA methyltransferase among our potential synaptic genes. Until recently, a number of studies came out linking ALK-BH8 variants to rare cases of intellectual disability in, 4 families as of 2021. And so I am going to tell you about Kim Madhwani's project investigating the function of ALK-BH8, which to date has been studied only in the context of cancer in mammalian cells. Okay, so Kim found that ALK BH8, that loss of ALK BH8 leads to an increase in the number of synapses. So this is a Drosophila NMJ. You probably heard a lot about this from the Littleton lab, or are in the Littleton lab and look at these all the time. But for those who haven't, this is the axon bundle, a single axon peeling off and then forming these boutons, each of which contains about a dozen synapses labeled here in green. And you can see that there are more boutons in ALK BH8 mutants, and that we can rescue this phenotype by restoring ALK BH8 function under the control of its endogenous promoter. And so we wanted to know what ALK BH8 was doing. So there are these two paralogs. Is ALK BH8 carrying out the canonical role of yeast TRIM9 and methylating wobble uridines? And so this is the step downstream of the elongator complex, which adds one methyl group, TRIM9 in yeast, with its obligate cofactor, TRIM112, adds a second methyl group at the 5 position of the base. There are additional downstream modifications that do occur subsequently, but I'm going to focus on this modification today. And what we find is that in ALK BH8 mutants, this modification is lost, as well as the downstream modifications. Okay? So it turns out that only a single tRNA has this terminal modification in animals, and that is tRNA selenocysteine. So we were intrigued by that. Selenocysteines decode UGAs instead of S-stop codons to incorporate selenocysteine and generate selenoproteins. And selenoproteins are well known for their role in regulating oxidative stress. And we had noted that oxidative stress has also been linked to synaptic overgrowth in Drosophila in particular. And so this is an example from the Sweeney lab where SOD1 mutants lead to mutations in SOD1, which adversely affects response to oxidative stress, leads to synaptic overgrowth. And a possible mechanism for this is through increased autophagy, but it's not known exactly why this is happening, and that's something that we're thinking a lot about, and I'd love to hear your thoughts. But so what we wanted to do was we wanted to see, okay, well, if we lose selenoproteins in another way, a completely independent way, do we see synaptic overgrowth? And this is, this gene, SPS2, is the selenophosphate synthetase. And so without it, you don't have selenoproteins either. And you can see, whoops, that we get, whoa, sorry guys. I've done funky things with my, my slides here. Sorry. I'm borrowing slides and not animating properly. I think I took slides that were working and made them not work. At any rate, this is overgrown, you can see, and you're just unfortunately going to have to take my word. It's very similar to ALK BH8. And then when we look at double mutants, we can see that they're overgrown to a similar degree, suggesting that they act in a linear pathway. So consistent with the model that ALK BH8 is methylating the wobble uridine in the tRNA selenocysteine, and this is required for the incorporation of selenocysteine into growing proteins, and therefore selenoprotein synthesis, and leading to synaptic overgrowth. Okay, so if oxidative stress is the root cause of the synaptic overgrowth that we observe in ALK BH8 mutants, we hypothesize that perhaps treatment with antioxidants would suppress our phenotype. And so again, you can see here that ALK-BH8 mutant synapses are overgrown. When we add the antioxidant AD4 or rear wild type animals on the antioxidant AD4, it doesn't change synapse number. But when we rear ALK-BH8 mutants, we see a partial suppression of the synaptic overgrowth. So not a complete suppression, but some suppression of the synaptic overgrowth, which was encouraging. And so we wanted to go on and see if there were downstream effects of this abnormal synapse formation that might tell us something about the link between ALK-BH8 and intellectual disability. And so we used a robust behavioral assay in flies that was developed in the Alex Keene and Kristen Scott's labs. And this is basically an aversive taste assay. And so what we do in this assay is you do a pretest, first of all, to make sure that the fly is hungry, so that it's been starved, and will extend its proboscis when presented with fructose. So flies extend their proboscis when they think they're being presented with food. They have taste receptors on their legs, on their tarsi, and so you can present them with fructose on their tarsi, and they will extend their proboscis. And so here we do this 3 times, and instead of getting fructose as they're expecting based on their tarsi, they get quinine, a bitter tastant that they don't like, okay? And then 5 minutes later, you test to see if they remember this negative association and do not extend their proboscis when presented with fructose to their tarsi. And what you can see is that wild type animals remember. They don't fall for it again. They're not extending their proboscis. But ALK-BH8 mutants do not remember at all. And so they are expecting fructose again every time. And so we can rescue this when we restore ALK-BH8. So this tells us that ALK-BH8 is important for learning and memory in flies. And so we were really excited by the possibility that antioxidant treatment could have a positive impact on this phenotype. And so, I should have said that this work was done by an amazing undergraduate in our lab, Shanze Saeed. And this work was done by another undergraduate, both working closely with Kim Madhwani, Carlson Ogata. And so this summer, he was able to do experiments where he reared flies on antioxidant and then repeated these learning and memory assays and found that we actually see restoration to wild type levels of this aversive taste memory when ALK-BH8 animals are reared in the presence of antioxidant. And so we're super excited about this. And so I sort of went through quickly. We have some more data supporting this model, but I wanted to just hit the highlights. And so our current model is that ALK-BH8 methylates wobble uridines in tRNA selenocysteine. This is critical for selenocysteine incorporation into growing polypeptides and selenoprotein synthesis. Selenoproteins then block oxidative stress, leading to proper synaptic growth and learning and memory. And in the absence of ALK-BH8, these processes are disrupted. And so the big question that we're trying to answer now is, can antioxidant treatment post-development? So we reared flies in antioxidant. So, flies are born and live out their lives in a little vial. And so these flies were exposed to antioxidant from, their parents were, and then they were as soon as they started eating in their environment. And so what we're wondering is if we can take adult flies and that have developed under normal conditions and expose them to antioxidant and see if we get any rescue of the phenotype. And so we're really hopeful about this because, as I mentioned, there are families and there are children living with intellectual disability due to presumably what appear to be loss of function mutations in ALK-BH8. Okay. I'm going to switch gears now. Does anyone want to ask me any questions about this one? Yeah. Looking at synapse number and structure and things like that? Yeah. That got loud. No, we haven't yet, but we are planning to, and yeah.
How many selenoproteins are there?
Very good question. Yeah, I sort of jumped over that a little bit fast at that point, sorry. So there are 25 in humans, and there are 3 in flies.
Can you rescue your phenotype by putting one of those?
By putting one of them, well, so we can recapitulate it by getting rid of all of them. We have not tried rescuing by putting all 3 back. That's a good idea. We have not done that. Yeah. Very good question. So I started out with the pattern and that we found its paralog, TRIMT9B, in the screen. That one is almost exclusively in neurons. It's so it's highly neuron enriched. This paralog, ALK-BH8, is it's expressed in every cell. That's correct. So the intellectual disability in humans is syndromic. It's got some other structural phenotypes, morphological phenotypes, but for the most part, the predominant phenotype is intellectual disability. And in our case, I have to admit that it's the one we've looked at is, we've looked at the brain. We don't see obvious deficits in other tissues, but we haven't looked deeply. Yeah. So it seems that may well be the case. Yeah. So that is our model, and I think it fits with, things like Alzheimer's and other things that where I think that ALS, oxidative stress, is particularly difficult challenging for the nervous system. Okay. And so the cool thing is we've actually now started studying others, and there are a number of others, other tRNA methyltransferase families, members of this expanded animal family of tRNA methyltransferases that are linked to intellectual disability. And so we're also doing a lot of proteomics and transcriptomics to identify other potential translational targets of these genes. All right, so I'm going to come back to this pattern. And so for the second part of my talk, I want to talk about this pattern. And so we were really intrigued by the fact that these synaptic genes had this really tight co-regulation. So they're roughly off at the same time, on at the same time, off at the same time, on at the same time. And this is many synaptic genes, hundreds of synaptic genes. And so we teamed up with Erica Larson, who is a genomicist at Brown University who studies coordinated gene regulation in early embryos. So she's interested in, among other things, X chromosome inactivation. And James Kentro is a joint graduate student in both of our labs. He joined the lab to do a rotation on the very first day we shut down for COVID. So we're very fortunate that he chose to join. And so, the nervous system is beautiful for its complexity, right? For its diversity. Many people are focused on understanding how this diversity arises. And yet, at the same time, all neurons form synapses. So they have shared features that make them neurons. And synapses are one of those components. And we know really almost nothing about how these the gene expression to build these common features of neurons is coordinated, let alone how it's carried out at the same time that the neurons are diversifying their fates. And so this is especially interesting to think about, I think, in the context of coordinating development of shared traits with neuronal diversification. So to make a synapse, you have to coordinately express shared synaptic genes, so synaptotagmin, complexin, calcium channels, but then also subtype specific genes, V-GLUT, VGAT, different receptors. You have to coordinate this between two cells. So you really need a lot of genes coordinately being turned on at the same time. And we were initially quite so let me just say this first. And we know that's happening. So I can break down this expression pattern to pan-neuronal synaptic genes versus subtype-specific synaptic genes, and we see this same expression pattern, okay? So they're definitely being coordinately regulated. And so we were really surprised because what little work has been done on this has been done in Oliver Hobart's lab, and he has mostly looked at diversification. And what he's found is that is this important role for terminal selector genes. So there are individual transcription factors in different neuronal subtypes that specify that neuron's fate. But then they did a study of about 30 pan-neuronal genes, so not specifically synaptic, though many of them were synaptic, to see how those were regulated, and found that it was totally different, a totally different regulatory paradigm with different transcription, combinations of transcription factors able to drive expression, none absolutely required to drive expression, and different ones in different cell types. And so we thought, okay, well, now the problem just got this much bigger. How are these genes being coordinately regulated at the same time? They have to be turned on at the same time, or you can't build a synapse. How is this happening? And so we decided to set out to try to identify regulators of coordinated synaptic gene expression. And so we reasoned that candidate regulators should have DNA binding motifs that are enriched at synaptic promoters and enhancers, should exhibit enriched binding at synaptic genes, should differentially regulate synaptic gene expression, and positively or negatively regulate synaptogenesis. And so this was our model that we started out with in our hunt for such proteins. And so James started during the pandemic while he was away from the lab, started with doing motif analysis to identify motifs enriched in our synaptic gene set, then analyzing existing ChIP-seq data to identify transcription factors who actually bound at synaptic genes. And so first was this motif analysis using tools like Homer and MemeSuite. And what he found was a lot of genes. So as many of you probably know, motifs are a dime a dozen. It's not the presence of the motif, but whether or not the motif is bound that's critical. But so this is a first step. And so the number one motif that we found is in a gene called DEF1. It turns out DEF1 is linked to autoimmune diseases and intellectual disability in humans. It's a DNA binding protein that also binds chromatin regulatory enzymes and can bring them to specific DNA sequences. So we'll come back to that. So then wanted to go one step further and identify actual binding at these motifs. And so this is where we were sort of at the limit of what's published. 173 of the candidates that came out of the motif search had published ChIP-seq data. And James also analyzed a bunch of other transcription factors that had published ChIP-seq data. He identified 11, so we really narrowed it down here. 11 that show enrichment at synaptic genes, and transcription factors with known chromatin remodeling ability are overrepresented. So what you can see here is this is DEF1 binding to synaptic genes, the level of binding to synaptic genes versus to all other genes in the genome, or upstream of. All right, and so the other, a couple other molecules that we identified that are also chromatin regulators are GAF and CLAMP. And so these are pioneer factors. They bind to condensed chromatin and can open it up for other transcription factors to bind. They also bring other modifying, chromatin modifying enzymes with them. They have been shown to interact genetically, both synergistically and competitively, depending on context. GAF and DEF1 also interact genetically, and DEF1 and CLAMP actually physically bind one another. And so so this is just a schematic of what pioneer transcription factors can do to open up closed chromatin and allow other transcription factors to come in. And so we came up with a model that DEF1 and CLAMP function together to repress synaptic gene expression. And so they would be acting at these troughs of expression. They would be binding and blocking transcription, whereas GAF then competes with CLAMP and promotes synaptic gene expression at these peaks of expression. And so this model predicts that chromatin accessibility at synaptic genes should be temporally regulated over development, right? So this implies that we're getting closed and open chromatin depending on over time. And indeed, that's what we see. So this is just one example of an acetylcholine receptor. And so you can see that in early stages, so this is based on our analysis of data from Eileen Furlong's lab, DNA-seq data, so showing chromatin accessibility. You can see that at 4 to 6, 6 to 8, and 8 to 10, this area is relatively closed. It's now accessible at 10 to 12 hours, which is just before we start to see the peak of expression of synaptic genes. And then we can do this for all the genes in our data set, and you can see here that other genes, background genes, which is every gene in the genome, that's not a candidate synaptic gene, is open, neurons starting to, synaptic genes starting to catch up, and by 10 to 12 hours, it's synaptic genes that are open. And so, our model also predicts that that the members of our candidate gene regulatory network will occupy these temporally regulated sites of chromatin accessibility, and indeed they do. So this is ChIP-seq data showing binding of DEF1, GAF, and CLAMP at these temporally regulated sites that I just showed you on the last slide. And so, we got this, and now, so this kind of puts them in the right place at the right time to be regulating synaptic gene expression, but are they regulating synaptic gene expression? So what we need to do now is have knockout models and do RNA-seq analysis to see if we're altering synaptic gene expression, and then go in vivo and look to see if we're altering synapse number. And so we've done that here for CLAMP. So this is RNA-seq of CLAMP null third instar larval CNS. And what you can see is in blue are the synaptic genes. And you can see that a large number of the synaptic genes, 60, about 2/3, are significantly upregulated in clamp nulls, consistent with it functioning as a negative regulator of expression, whereas almost none are downregulated. And in fact, when we do a GO analysis of all of the upregulated genes in clamp nulls, we get, ion transport, cell communication, signaling, synaptic signaling, chemical synaptic transmission. So it really seems as though we have identified a negative regulator of synaptic gene expression in clamp. So what does it mean then if you fail to appropriately negatively regulate synaptic gene expression? What does that mean for synaptic development? It turns out that you do indeed form more synapses. And so in a clamp loss of function model, we see significant synaptic overgrowth, consistent with our model that clamp is negatively regulating synaptic gene expression and synapse formation. Similarly, loss of DEF1. So that is not supposed to say clamp, that is supposed to say DEF1. And so is that. Sorry about that. This is also significantly overgrown. So loss of DEF1. So we're in the process of doing the RNA-seq, so I can't tell you yet about the regulation of synaptic genes in DEF1 mutants, but I can tell you that we do see increased synaptogenesis. And so back to our model. So these results are consistent with this model. And the ongoing experiments are RNA-seq, and we're now doing a TAC-seq too to try to get an idea of chromatin accessibility in both clamp and DEF1 mutants. We are investigating the co-localization of clamp and DEF1 on polytene chromosomes. So we would expect that we would see them together. And then we are assessing synaptic growth, gene expression, and chromatin accessibility in the absence of GAF. This has been a little bit more challenging. Clamp and GAF have very early functions in the embryo. They're involved in the maternal to zygotic transition of gene expression. And so finding good ways to knock them down effectively while allowing them to survive to larval stages has been a bit of a challenge, particularly for GAF. And so the next question that we're trying to address is about whether or not this candidate gene regulatory network that we've identified, or even this mechanism where, so basically what we're hypothesizing is that upstream of these two different paradigms that the Hobart lab has found for regulating the expression of subtype specific versus pan-neuronal genes is chromatin accessibility, is the regulation of chromatin accessibility. And so they're co-regulated at that level. Is that true in all neuronal subtypes? Is the same gene regulatory network responsible for regulating chromatin in all neuronal subtypes? And so to look at this, we have been collaborating with Jay Shandur and Eileen Furlong, who did this, just this monumental amount of work, making 2-hour time points, overlapping time points across development, single cell, attack, and RNA-seq data sets. And so our role was to annotate the neuronal data sets. And so you can see here that we can narrow these, we can get pretty decent clustering, and we can identify different types of neurons in these based on the expression of genes known to be exclusively or predominantly expressed in specific neuronal subtypes. And so that should allow us to do this analysis. We can look at synaptic gene expression levels, so we can overlay this over the clusters, just the level of expression of our list of synaptic genes. And you can see that the expression is highest here. And that correlates with the older cells. And so that makes sense. This is non-neuronal. This is actually head mesodermin. So that makes sense. And in fact, when we basically take these single cells and then we do pseudo-bulk analysis, so we put them all back together, we can recapitulate, we see the same rise in synaptic gene expression occurring between 10 and 12 hours. Now this data set ends here, so we don't have the dip, but suggesting that this data set is going to be very useful for us, so reinforcing the idea that these are coordinately regulated and allowing us to look at different neuronal subtypes to see how synaptic gene expression is coordinated in those cells. And so that's the plan. So we will look at our candidate gene regulatory network across neuronal subtypes. And then we're also hoping that we can use this data to identify new gene regulatory networks that may have been obscured in the bulk data. So in the bulk data, if there's something that's happening in one subtype of neuron, you know, for example, peptidergic neurons, we may not see that. And so we're hopeful that we'll be able to flesh out this analysis and understand more about how synaptic genes are regulated. We're also starting to collaborate with some mouse labs, with some brain organoid labs to try to see if and how synaptic gene expression is coordinated in those contexts as well. And so with that, I would like to, again, thank the lab. Again, the work that I talked about was done by Kim Madhwani, the ALK-BH8 story. James Kentro worked on the coordinated synaptic gene expression. Our collaborators, Dragani Fu and Jenna Lentini, whose names I think I failed to say, who did the tRNA mass spec and our tRNA expert collaborators that we're thrilled to be working with. Erica Larson, expert genomicist, and our funding sources and the graduate programs at Brown that I'm associated with. And this is a picture of Waterfire. I got here on the train in 30 minutes. You could come down, should come down sometime and visit us and go to Waterfire. All right, thanks. Yeah. Mm-hmm. Right. 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 So that's a very good question. So one thing to remember is in Drosophila embryogenesis lasts 24 hours. So everything happens very fast and kind of all at once. And so the timing precedes synaptogenesis by a bit. The details of whether or not synaptogenesis is occurring at the same time in all neuronal subtypes are not like systematically worked out, and the answer is probably no, they're not. And so one thing that we're hypothesizing is that there's potentially a second layer of regulation at the level of translation. That's correct. Temporal expression for one of those peaks. Yes. Oh. Yeah. I see, I see. So you're saying that maybe they're differentially regulated at the different developmental times. That's entirely possible. And actually, there is one thing that we have done that's akin to that, but not exactly that. Let's see if I can just get this pattern back up. Well, here's, I'll just go to this one. This is a simplified version of it. So there are actually two subclasses within here, one that has a lower peak early and a higher peak later, and vice versa. And so we have analyzed those separately. Yeah. Yeah. Yes. Right. Yeah. I think that clamp and GAF are general factors. I think the specificity is coming from DEF1, but we don't know that yet. So DEF1 is neuronally enriched. It's expressed in, it's expressed much less broadly. And we can look at the single cell data and we can see that DEF1 motifs across embryogenesis are only accessible in neurons.
So if you understand and clamp or DEF1 mutants for synaptic proteins, do you see more synaptic proteins, or is it just something that's making more glucons? Or?
Right. So we have not, rigorously quantified the protein level, like per synapse, and then, extrapolated from total synapses. We're just operating right now on the assumption that each synapse has a normal complement of protein, but it is possible that that's not true.
Are these animals lethal?
Yes. In null mutations in these genes are lethal. Yeah. Right. Right, 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 right. Yes, and so that is something that we're in the process of doing. And so yes, again, so yeah, we're kind of acting on the assumption that there are more active zones and that there's more protein, and those are things that we're actively looking at right now. Yeah. Right, right. That's not the case at the Drosophila NMJ. Yeah.
So, it's just that because of the connection of the clamp to the central center, any chance that you can also regulate, receptors that most patients
Yeah. So it's very interesting to think about whether or not the clamp may be playing a role in sex-specific differences in the brain, and that's something that actually a postdoc in Erica's lab is actively looking at, and that we're thinking about in the context of synaptic genes. We've been using both. Yeah. Yeah, but we are for the RNA and ATAC-seq that we're doing. We're planning to do both separately, so we can try to parse that out if there's anything there. Yeah, it's a really good question.
What actually goes on in the first part of your talk when you get rid of your methyltransferases?
Yes.
Wobble biology doesn't happen, and you don't insert that specific cysteine. Get a stop going on, or just some other amino acid happens? What is happening to these proteins?
stop. It doesn't get recoded.
So the stop codon.
So it's a stop
Indicated
codon.
protein, are these early, where they are in the protein?
Yes, and so so selenoproteins are really interesting. I did not know this. Most, if not all, selenoproteins have one selenocysteine, and they, I think in many cases, certainly in Drosophila, have a paralog that's not a selenoprotein. So that has a cysteine, or in some cases has a stop that doesn't get recoded. And so there's a sequence in the 3' UTR that's required for the recoding of the internal UGAs, as well as the methylated tRNA selenocysteine. And so if you don't have both of those things, so that's kind of how you can computationally fish them out, is with the sequence in the 3' UTR and then the internal UGA. If you don't have both of those things, you will not incorporate selenocysteine. Does that answer your question?
And these are typically in terms of where they are located.
Where they are located, I don't know if there's a typical place where they're located, like late or early in the molecule. I don't know that.
Any final questions before we go to the reception? If not, the reception on the atrium. And let's take one more time.
Thank you.